Want to get off. Remember that song? Randy Stonehill.
So bad because analytical comes so naturally to me, but that exam is. Well, I guess it's same as I have my homework for you. Didn't have a stapler, so all the extra sheets are just kind of tucked in.
Okay.
Just so you know.
Hello. Hello.
Hi, how are you doing?
I'm testing my microphone. Yeah, it looks like I found one that worked. So tough break, huh?
Yep.
I can't even imagine what you were all going through.
Yeah, it was pretty upsetting. And the person who started it wasn't even on the trip. He just gave it to somebody that was.
Oh, good golly. So sorry to hear
One
that.
person made.
Yeah.
It is.
At least I think so.
Okay. Did you guys get the email I sent you?
About lab, yeah.
5 seconds ago.
No, yesterday I sent it the picture from the Western.
Yes.
It worked. Not only worked, it worked well. Beautiful band, relatively clean, really nice and clean. There's a faint band next door where the whole extract was done, but the concentrated, nice, clear, beautiful, clean band. Good experiment. Good job, everybody. Pat yourselves on the back, but not too hard. So
are we officially done then?
except for your projects, yes.
Well, we can write the lab report now.
You can write the lab report now, and the three of you can work on it together. Dane, it was a very long day. We started at 8:30. We got done about 4 o'clock.
Oh,
So, but we got it done. And
that's
it worked.
good.
So
Also, I think I need help doing.
didn't grow at all.
I don't know what I did. I even checked it with the absorbents just to see if I just couldn't see it. I have no idea what I did.
Let it keep growing. It may take longer to
I put it
grow.
back in the shaker,
Yeah,
but-
just let it go. Or you have your original culture that you gram stained.
Mhm.
Maybe restart and just use more inoculum, something
Right,
like
'cause
that.
one thing that I thought is because you had restricted the day before, like there weren't amazing colonies yet, and they were kind of small, but now there's.
That's okay. Small colonies are no big deal.
Because there's better colonies now, so I'm wondering if maybe it just.
Difficult to say.
Sometimes that like, it's like, I think I can, you can or not. It's just the problem is I don't have time.
All right. Just a word. Oh, by the way, I don't know if you guys know Audrey Morris. She's in the nursing program here. Her father died last night of COVID-19. Left 4 kids.
Oh.
And Audrey's the oldest daughter. So being the oldest daughter, it's kind of fallen to her to take care of the other 2 daughters. She has a 17-year-old who's pretty self-sufficient, but a 4-year-old who's pretty demanding. And her mom is recovering from a pretty severe bout of COVID-19. So I cannot even imagine what she's going through. So, so let's keep the Morrises in mind today. And then the soccer team. To work that hard to get there and then to just be told, go home. Because a couple of you are sick. Yeah, anyway. Then that insult to injury, you have to quarantine. So, all right. Let's pray. Father, thank you for this day. Thanks, a little warmer, even though that wind is still freaking cold. Thank you that soccer team's back safely, and we pray for the Morris family. Lift them up. Thank you that she has, Audrey has a great cluster of friends, both in the nursing program and at admissions where she works. Lord, be their light, their salvation. Remember that you said you are the light of the world. Whoever follows you will never walk in darkness, but will have the light of life. And be the light of their lives now. And now, Lord, help us to learn the rest of this stuff about purines and pyrimidines and their synthesis and degradation. In Jesus' name, amen. Just a word about the tetrahydrofolate reductase. In protozoa, thymidine kinase and tetrahydrofolate and dihydrofolate reductase exist as a bifunctional enzyme. For that reason, several of the drugs that are used to treat protozoan infections will target that bifunctional enzyme, and they work quite well. So that's a target that does not exist in mammalian cells because of that. So that's something to keep in mind. Without thymine, cells cannot make DNA. So this death without thymine is called thymineless death. In fact, bacteria, one of the ways you can kill cells that aren't, if you want cells that are in stationary phase that don't grow, is you put a mutation in the enzyme that makes thymidine. So the thymidine synthase, thymidylate synthase, and then you deny them thymine. And those cells that want to keep growing will undergo thymineless death. Rapidly metabolizing cancers are very susceptible to thymineless death, and a very powerful inhibitor of thymidylate synthase is a drug called 5-fluoro-2-deoxyuridine. And that drug is a active site inhibitor. It reacts with amino acids in the active site to form a covalent adduct with the enzyme, thereby inactivating it. So this is a Achilles heel for fast-growing cancers. No, it's cold. This side of the building, I don't know what it is. It just, the radiators are supposed to be plugged because the water, Spring Harbor water is terrible. And this building doesn't have any water treatment. And I'm not going to get on that hobby horse because I've shot that horse dead. I've beaten it so many times. Okay, so cells can make new nucleotides just from scratch. That's de novo synthesis. They can also make them from recycled purines and pyrimidines, and that is called salvage synthesis. And these are salvage pathways. They use the products of nucleotide degradation to make new nucleotides. These pathways are relatively straightforward, but they're very heavily used by cells. So they're important. So free adenine can react with PRPP, with the ribose 5-phosphate 1' pyrophosphate, PRPP. This enzyme is called adenosine phosphorybosyl transferase, or APRTase. And the adenosine phosphorybosyl transferase takes PRPP and adenine and converts it to AMP and inorganic phosphate. Then the inorganic phosphate is degraded by inorganic pyrophosphatase to make two molecules of inorganic phosphate that drives the delta G of the reaction further into the negative realm. This enzyme gets a lot of use. So this is how you make AMP. AMP. For guanine, you can use hypoxanthine guanine phosphoribosyl transferase. This is called HGPRTase. And this enzyme will catalyze either one of these reactions you see here. So it can use either hypoxanthine or it can use guanine. And if it uses hypoxanthine, it makes the inosine monophosphate. Inosine monophosphate can be used to make either ATP, AMP, adenylate, or GMP, guanylate. Consequently, this pathway, this enzyme, is controlled by products of this of those diverging pathways. If you use guanine, you just go straight to guanylate. So the products of this, GMP, IMP, they inhibit the HGPRTase, hypoxanthine guanine phosphoribosyl transferase. The adenosine phosphoribosyl transferase is inhibited by AMP, and then AMP also inhibits the adenylosuccinate synthetase. Guanine will inhibit the inosine monophosphate or the anodylate dehydrogenase. And so we can take the regulation of this, draw it this way. Here are your salvage pathways here. So this is catalyzed by the adenosine phosphoribosyl transferase. This is catalyzed by the hypoxanthine guanine phosphoribosyl transferase. The end products of these both. So this end product here and this end product here, both feedback inhibit their enzymes, as does this one here. This one here will inhibit the IMP dehydrogenase, and then AMP also will inhibit this enzyme here. That's the adenylosuccinate synthetase. Adenylosuccinate synthetase. And so these pathways, you remember from the de novo pathway, also guanine is converted to guanylate, and that is also inhibited by guanylate as well. So the end products of these enzymes inhibit the actual enzyme itself, as do these to the de novo pathway as well. That way, you don't overuse the de novo pathway when you have the salvage pathway going. This is a great way for cells to save ATP, because ATP is at a premium, and these pathways use a lot of it. Now, inherited mutations in hypoxanthine guanine phosphoribosyl transferase. That gene is X-linked. Mutations in this gene cause a very interesting genetic disorder called Lesch-Nyhan syndrome. Lesch-Nyhan syndrome.
Did we talk about this before?
We might have. So this X-linked genetic disorder will cause aggressive behavior, mental retardation, poor coordination, and a marked propensity towards self-mutilation. In particular, the patients with Lesch-Nyhan syndrome chew their lips, and their lips actually will fall off because they chew them so much. Now, why do they do this? This is still mysterious, but The guess is that the brain is very dependent upon salvage pathways for the synthesis of its nucleotides. Without those salvage pathways, you get a buildup of uric acid. And the uric acid damages the central nervous system, and it's clear that people with Leese-Ninehan syndrome have increased uric acid in the brain. In mammals, salvage of pyrimidines is not as important. When it does occur, it occurs through nucleosides. So with purines, it's the bases, the free bases. They're not attached to anything. With the pyrimidines, it's the nucleosides. So uracil and ribose 1-phosphate. Are converted to uridine. Uridine, remember, is the nucleoside. And inorganic phosphate. The enzyme is uridine phosphorylase. Uridine phosphorylase. And it catalyzes this reaction here. So that is an enzyme that is pretty widely distributed. Now I have uridine. Uridine and ATP will make uridylate and adenosine diphosphate. And that is uridine kinase. We've seen uridine kinase already. Now the mammalian uridine phosphorylase, this enzyme here, has an increased substrate allowance, shall we say. So it will use not only ribose 1-phosphate, it will also use 2-deoxyuridine as a substrate. So instead of getting uridine, you get 2-deoxyuridine with inorganic phosphate. So the mammalian uridine phosphorylase will use either the ribose 1-phosphate or use 2-deoxyribose 1-phosphate. Thymidine. Thymidine has a very similar pathway that it uses. You use thymidine phosphorylase, catalyzes the same reaction, free thymine and ribose 1-phosphate, except it's not ribose 1-phosphate, it's deoxyribose 1-phosphate. That gives you thymine plus the inorganic phosphate, then thymidine plus ATP gives you deoxy-TMP or deoxythymidylate and ADP. And these enzymes, in terms of their 3D structure, are very similar. This enzyme here, uridine kinase, and this enzyme here, thymine kinase. Very similar. What about cytidine? Deoxycytidine kinase will convert the free deoxycytidine, so that's a nucleoside, to deoxy-CMP with the subsequent hydrolysis of ATP. That's how you get the phosphate. And I'm not sure why.
The highlights there.
Huh. Well, that reaction was there, but you can't see it. Okay. So this shows you some of the conversions and interconversions. You have interconversions of pyrimidines. These pathways, as you can see, are very highly regulated. Also notice that there's cross-regulation as well. Take a look at how the deoxy TTP regulates the conversion of deoxy CMP to deoxy UMP. That's essentially the first step. It also inhibits deoxy TTP, inhibits the thymidine phosphorylase. That step. Then it also inhibits a step all the way up here. That converts the the UDP to deoxy UDP. That, of course, would be what enzyme? We talked about that last time. Ribonucleotide reductase. So is that. That's ribonucleotide reductase. Ribonucleotide reductase, RER.
Okay.
And then these interconversions, as they can occur, they are they're cross inhibited. This one negatively inhibits that. That one positively regulates that. So on and so forth. Blah, 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 what have you. So DCTP, DTTP, they're the major positive, negative regulators of the interconversions of these salvage reactions. Now let's talk about nucleotide degradation, if you're all good on salvage pathways. So purine degradation flows into a common pathway. So we know this common pathway, is the pathway by which you degrade all of your purines, essentially. So adenylate, guanylate, the starting products of purine degradation. And here's essentially how they work. So the enzyme adenosine deaminase is going to convert adenosine to inosine. Both of these, first of all, are stripped of their stripped of their phosphate. So you have a 5' nucleotidase. That's really just a phosphatase, but specific to nucleotides. Then you have a nucleosidase that will essentially liberate ribose, and you get hypoxanthine and guanine. Hypoxanthine and guanine. So notice these enzymes here are the same. The intervening enzyme here is adenosine deaminase.
Oh, thank you. That's a lot better.
Okay. So this here is the intervening step that's not shared by these. So you convert adenosine into inosine. Adenosine is converted into inosine. By converting adenosine to inosine, now you're going to get hypoxanthine. Okay. Both of these then are going to be converted to xanthine. And this is the enzyme xanthine oxidase or guanine deaminase, either one. The guanine deaminase for guanine, xanthine oxidase for hypoxanthine. Xanthine oxidase can also use xanthine as a substrate. It'll convert it into the end product here, uric acid. Notice this is a two-step reaction. So in this case, both steps are catalyzed by xanthine oxidase. Here, the first step is converted by guanine deaminase. Second step is converted by xanthine oxidase. Xanthine oxidase is a very heavily studied enzyme. It contains 2 iron sulfur clusters, a bound FAD cofactor, and also a molybdenum. Then you get uric acid. Uric acid is not that soluble. Xanthine is more soluble. Why do we convert it into uric acid? Uric acid turns out to be neuroprotective at low concentrations. So your body will actually reabsorb some of the uric acid. And so there's a low level of uric acid in your blood that seems to be neuroprotective. If you get too much, it will precipitate out at joints. It makes these things that look like little sewing needles. They do a lot of damage at joints. First joint that's usually affected is the big toe. And people with gout have told me it feels as though someone took an army knife, stabbed it into your toe, and it's just twisting it and just keeps twisting it.
Is there a reason that's the first joint to be affected? I feel like it would.
Probably because it's the most weight-bearing joint.
Huh.
Also, you have a big surface for precipitation there. You do at the knee as well, but the knee has a bigger bursa and is flushed better than the big toe.
Yeah, I would have assumed it would have been something like the knee or the hip.
Yeah. Now, notice other animals and creatures will use enzymes like urate oxidase to make allantoin. Allantoin is pretty soluble. A lot of mammals make allantoin. So primates, birds, reptiles, insects, they make uric acid. And uric acid, because it's not very soluble, you don't lose a lot of water when you excrete it. So that's one of the beauties of uric acid. In fact, birds, when they mess your car, that's why it etches your paint. Other mammals excrete allantoin. Then there is an allantoinase in bony fishes that make allantotate. That's quite soluble. Then this enzyme here, allantoicase. That's kind of a weird little enzyme, but it makes glyoxylate, which is also very soluble, and that makes urea. In cartilaginous fishes, you concentrate urea in your tissue, that helps with buoyancy, along with squalene. Others use urease to just make ammonium, and if you're marine and you have gills, big deal, you just excrete it out your gills. Other creatures stay away from ammonium because of the dangers of ammonia to metabolism. It poisons the Krebs cycle. It removes alpha-ketoglutarate, it removes oxaloacetate, and poisons the Krebs cycle. Now, mutations in the gene that encodes adenosine deaminase, that enzyme right here catalyzes this reaction, prevent your immune cells from making DNA, because they use that degradation to actually fund their salvage pathway. That's how these cells, when say you get the COVID-19 vaccine, like I'm going to get later today, that's how they expand. Without this enzyme, you can't expand, and you have a condition called SCID, severe combined immune deficiency. You have to live your life in a bubble because you're subjected to massive infections of just everyday germs due to mutations in that gene. So those mutations also prevent the breakdown of deoxy-ATP. High levels of deoxy-ATP inhibit ribonucleotide reductase. So now you can't make DNA because of the regulation. And as we said, buildup of uric acid, overproduction of uric acid will cause gout. So the exact cause of gout is not well known. It is clear that diabetics are at a higher risk for gout. So It seems as though blood sugar regulation is linked to uric acid synthesis and disposal, and so on and so forth. Beyond that, nobody really knows what causes gout. Now, is gout, does it run in families? Yes, it does run in families. But it's clear diabetics have a larger risk for gout. If you look at the statistics, diabetics suffer from gout at a much higher rate. And it's mainly type 2 diabetics, not so much type 1. However, you get type 1s with poor control, who don't take care of themselves, yes, they can get gout.
Along with a slew of other things.
Yeah, along with a slew of other things as well. So, how do you treat gout? You use an inhibitor of xanthine oxidase. This inhibitor of xanthine oxidase will create xanthine and hypoxanthine that are much more soluble. So you excrete them, you don't make as much uric acid, so you have fewer crystals. Is that your Dexcom system? Does that mean you're doing really well, or?
No, it means my blood sugar is going up. If it's like an up tone, that means it's going up. If it goes, then it's going down.
Oh, okay.
And then if it's a high pitch beeping, I'm high. If it's a low pitch beeping, I'm low. And then if it sounds like a tornado siren, then I'm very low.
Woke me up a little
Okay.
bit.
Lisa doesn't talk to you.
You can change them to be different.
Your
tones.
blood sugar is low. You're such an idiot. You know,
Yeah,
just,
most people aren't attuned to like the different.
your blood sugar is too high. You're so undisciplined. I'm very disappointed in you.
Sounds like you sound like my old doctor.
Yeah.
It's funny that after 4 years of being around you, it doesn't phase you that it goes up anymore.
Yeah, it goes up all the time.
Okay. So the allopurinol is converted to the oxypurinol. As you can see, that's a very strong inhibitor of the xanthine oxidase. There's also another drug that's an inhibitor of xanthine oxidase called Febustat. Febustat's a newer drug, but even though it's newer, allopurinol is still a better drug, it seems. And these are the other parts of the pathway we talked about. I won't belabor that. Yeah, gout would.
So does it get sharp because of the buildup of crystals? Or is it?
The
like
crystals
one
just the way they're
needle?
formed. They're like little needles.
Okay. So is each individual crystal like one needle?
Yeah, and you get bunches of them. When you see x-rays, in fact,
So what are the other symptoms of gout?
Can you see them on the outside of your skin tissue, like bulging?
yeah,
I wonder what that looks like.
okay. Come on, go down.
Ouchie. Oh.
They took this out where it says view. Anyway, so if we click on that, that's Wikipedia. And yeah, it's Radiopedia. This is a great site if you want to. Yes, I'll accept your stupid cookies. You, go away. Don't want you anymore. Okay, so you can see the joint damage?
Is it almost separating the joint?
Yeah. And see what it does to the bone? You get chips of bone and cartilage in there. See, see, look at the damage here.
If it gets bad enough, would they amputate?
Yeah.
So what's the difference between gout and like a bunion?
Bunion is a bone because of the way the weight is placed on your joint. You get, so the joints remodel in response to increased force. So you get a remodeling and you get this big knob of bone here, and then this gets displaced this way. So this then the bone actually will do this. And then this gets big like that, and that's a bunion. And the doctor will laser that off, and then the doctor will have to reset. So the doctor actually has to break your toe, laser that off, and then reset it. They usually put in a bar so that it resets, and then the bar is taken out. And then there's a new joint that's actually made with a material that's put in.
Isn't bunions genetic?
Bunions do tend to run in families. Bunions also run in people who wear certain types of shoes. They, why is your finger in front of your camera?
Because I had you on my phone. I had just gotten back from being tested, so it was just the way I was holding. my
Oh,
phone.
okay. Was it a laminar flow test or is it PCR?
We do both. My antigen was, my card was negative, and then they sent my
Oh,
PCR.
okay. So that's good news.
Yeah.
So what are the other symptoms of gout?
The joint pain mainly. The inflammation that it causes can cause fever and headache and weakness and other types of things, but
'Cause I know they'll amputate diabetics, but due to.
that's due to gangrene.
Yeah.
Yeah.
I would say due to infection. from
Yeah.
circulation.
Yeah,
Does gout make you more susceptible to infection?
can, yeah. Especially if it starts to poke through the skin.
Oh,
It's got to get really bad for it to poke through the skin.
that just sounds terrible though.
Because it's in the joint capsule, it's got to essentially cut through, which can happen.
Right, but I guess for it to do that, would they have had to like go untreated for a while?
Yeah. Also, if you have gout, you have to be on a diet that decreases nucleotide intake. So you can't have yeast, can't have beer or bread or certain types of organ meats or things like that. And many people with gout just say, screw it. I'll eat what I want. And especially if they're diabetic and their blood sugars all out of whack too. And
They're already saying.
they're already, what have you. So, yeah. Okay. We will talk about gout a little later. Amanda, I sung with a guy in the chorale, Bill McMillan. Bill was in his 80s.
Some people are really well controlled.
Bill never had a blood sugar over 200, largely because one, he wasn't brittle, two, his wife nagged the crap out of him. And Bill, even in his 80s, had feeling in all his toes. He had no neuropathy in his feet.
I'm better than a lot of people, but I'm still not great.
Once you get better, once you can thrive.
Yeah, so once my hormones chill out, then it's a lot easier to manage.
You know what else usually helps diabetics? Getting married.
Well, that's not happening.
Because they have accountability.
Because now it's just not them. They have to be alive for someone else. They tend to take care of themselves. Just like driving rates, your insurance goes down when you get married, because now you drive a lot more carefully. Then you have kids, it goes down even more.
That's very fascinating.
Then your kids grow up and they start driving, then it skyrockets.
I do think that's fascinating because my grandma got divorced when I was really young and it kind of did the opposite. Where's the power and self-control?
Yeah, I like. It's just hard, and so it doesn't make it any easier to have like a mental illness on top of it.
Yeah.
That makes it so much harder to deal with. But I'm still, my doctors still give me a pat on the back and send me on my way. So,
You do what you can do. You can't be Supergirl,
and I also, if I have a high or a low, I'm not gonna take the joy out of life or stress myself. That's a big part of it. Is sometimes to be that well managed, you have to stress yourself out and monitor it to the point of like, well, what you have to have is you have to have a routine that is so strict that there's no room for anything. So you eat the same thing at the same time every single day.
Yeah,
And do everything at the same time. Yeah, it's boring and it's very monotonous and it sucks the joy out of everything.
Yeah.
To have perfect
Yeah.
blood sugar.
yeah.
So.
Well,
Even like
pyrimidines,
go to sleep at the same time.
so pyrimidines, you can see here, are degraded to bicarbonate and ammonium. So those aren't going to give you any trouble unless your liver fails. So these, as you can see, all of these here all get converted to uracil. Cytidine, 2-deoxycytidine, deoxycytidylate, 2-deoxyuridine, deoxyuridylate, all are converted to uracil. And here essentially is how that happens. So this enzyme here, cytidine aminohydrolase, very widely distributed enzyme. It's found in bacteria, it's found in plants, it's found in mammalian tissue. Then this enzyme here, the cytosine aminohydrolase, and this one here, that will convert cytosine to uracil. That is found in bacteria and yeast. The deoxycytidine aminohydrolase, also very highly distributed. That'll convert it to 2-deoxyuridine. And then deoxycytidine aminohydrolase also will convert DCMP to DUMP as well. So it doesn't do just the 2-deoxycytidine, also does the deoxycytidylate, converts it into deoxyuridylate. Then you have a uridine phosphorylase. The uridine phosphorylase essentially converts both of these to uracil. And you get ribose, deoxy-1-phosphate, deoxyribose-1-phosphate, ribose-1-phosphate. Those get recycled. Those can be recycled, no problem. Here you have a 5' nucleotidase. That should remind you of an enzyme. Here, 5' nucleotidase, yes, it's the same enzyme. And then all that. Okay, then uracil, as you see here, will be converted to dihydrouracil. So essentially you go the reverse from the way it was made. You're going to reduce it. So you have dihydrouracil. That's dihydrouracil dehydrogenase. Then you're going to put a water in, open the ring up. That gives you beta uretopropionate. Beta uretopropionate. And then your beta uretopropionase will make beta alanine and ammonium and bicarbonate. Thymine is also a substrate for dihydrouracil dehydrogenase, interestingly enough. And this essentially goes through those steps. You see those here. And it also gets converted to the beta ureto. Actually gets converted to beta ureto butyrate. And then this same enzyme is going to convert that to beta amino isobutyrate, which gets deaminated to make methylmalonyl semialdehyde. Methylmalonyl semialdehyde is an intermediate of valine catabolism that gets degraded ultimately to succinyl coenzyme 8. Cytidine degradation, again, we went through these enzymes, very widely distributed. Cytidine aminohydrolase. Cytosine aminohydrolase. That enzyme doesn't occur in humans, just occurs in yeast and bacteria. Deoxycytidine aminohydrolase, that does occur in humans, as does the deoxycytidylate aminohydrolase. You can see those here and here and here. And then uracil, we already went through that. Uridine phosphorylase, that gives you uridine, 5' nucleoside date, because you know, blah, 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 what have you. You can see that there. That will handle both uridine to deoxyuridine and to uracil, then the uracil gets degraded accordingly. And then thymine also gets degraded. You have the dihydrouracil dehydrogenase. You get dihydrothymine. Notice the enzymes are the same. Dihydropyridinase, that gives you beta uretobutyrate. Beta uretobutyrate. And then these two same enzymes give you the beta aminobutyrate. You deaminate that, you get the methylmalonyl semialdehyde. That gets converted to propionyl coenzyme A and succinyl coenzyme A. That gets degraded in the Krebs cycle. Okay. Nucleotide synthesis is an important target for anti-cancer drugs. You have to make nucleotides if you're going to grow quickly. Cancer cells grow quickly. They pride themselves on their ability to grow quickly, so to speak. So if you inhibit nucleotide synthesis in cancer cells, you'll kill them. So many anti-cancer drugs attack cancer at this vulnerability.
Would they cause like problems with other cells though by blocking?
Yeah. So all of your fast-growing tissues will be affected. Tissues in the stomach, hair follicles, bone marrow, skin.
So it's like chemo and so hard.
Exactly. If you use these types of drugs. So this group of anti-cancer drugs are called antimetabolites. They're going to hit drugs during the S phase of the cell cycle. That's the time when these cells are making DNA and RNA. You inhibit those enzymes, the cell can't grow, it dies. So the synthesis of deoxy-TMP, as we said before, is a big target of some of these drugs. So 5-fluorouracil. Here's the structure of 5-fluorouracil right here. You got a uracil, but you got that fluorine group up there. So it is converted into the fluorodeoxyuridylate, and it's converted into the fluorouridylate, the fluorouridine triphosphate. This is used to treat breast, head and neck, pancreatic, gastric, colon, rectal, esophageal, liver, genitourinary, that's bladder, penile, vulva, prostate cancers. There's that drug there. Then we have trimethoprim, which is actually an antibacterial, and then methotrexate. This is a hugely important anti-cancer drug. This drug is also used to treat certain types of autoimmune diseases. It's used to treat Crohn's disease, used to treat non-ulcerative colitis, doesn't work against ulcerative colitis, It's used in rheumatoid arthritis. Very important drug. So the 5-fluorouracil is converted to 5-fluorodeoxyuridylate that inhibits thymidylate synthase. And so this enzyme, as you can see here, I'm going to stay over here so Dane can hear me. When this binds, you have the tetrahydrofolate, and the tetrahydrofolate will give this methylene group here. You have withdrawal of these electrons here by these two nitrogens. Therefore, you'll have attack with this double bond of this carbon here because you generate a carbocation. You attack the methylene carbon here, you form a bond. In order to resolve, you have to steal, you have to abstract that hydrogen there. You methylate that just like that. Bob's your uncle, there you are. With the deoxyfluorouridylate, when you form that bond, this ends up as an electron sink. Because it's an electron sink, you don't resolve that because this is now stable. So this makes a dead end covalent complex. That's how this inhibits the thymidylate synthase. Methotrexate, along with aminopterin, which is used in the laboratory, not used so much in medicine, and trimethoprim, which is used in antibiotic, will inhibit dihydrofolate reductase. And because you don't regenerate the 7,8 dihydrofolate, this important cofactor cannot work. So that's two ways essentially to prevent cancer cells from growing. The electron sink is a very electronegative compound element. So those electrons go straight to the fluorine. Therefore, when you make that adduct, it's now stable because any excess electron density just gets pulled straight out to the fluorine, and fluorine's happy to have it. Whereas before, that's not the case. You're going to let go of that, and you're going to abstract now that carbon. You can't abstract that carbon now because you can't get a heavy enough need for it because those electrons have gone to the fluorine. Okay, so that's fluoxuridine. Fluoxuridine, that gets converted to 5-fluorouridine. That inhibits the uridine phosphorylase. Uridine phosphorylase. It interferes with DNA synthesis. It also gets incorporated into RNA. And then the monophosphate version of fluoxuridine, the futtermup as it gets called, that's easier than 5-fluoro-2-deoxyuridine 5-phosphate. That inhibits thymidylate synthase. This will inhibit several different enzymes. It's a very powerful anti-cancer drug. Tetrahydrofolate becomes a target as well. You inhibit that, you inhibit the synthesis of deoxythymidylate. For the reasons that we just talked about. Okay, so the serine hydroxymethyltransferase will regenerate tetrahydrofolate into N5N10 methylene tetrahydrofolate for its participation in the synthesis of deoxyuridylate. So methotrexate, aminopterin interfere with that. And then the deoxycytidine base, this so-called RACTP, known as cytarabine, This is incorporated into DNA because of strand termination. This will use arabinose instead of ribose. The polymerase can incorporate it, put it into DNA, but then it can't add on to it. So any DNA you make is terminated. This is the drug of choice, actually, for first round treatment for leukemias. Non-lymphocytic, lymphocytic, myelogenous, and chronic myelocytic leukemias, and then some other tumors, non-Hodgkin's lymphomas, and a certain type of rare tumor, leptomeningeal carcinomatosis. That's also treated with cytarabine. Then there's capecitabine. Capecitabine is an oral 5-FU prodrug. 5-fluorouracil prodrug is converted to 5-fluorouracil by liver and also by tumor cells. So this is used as an adjuvant treatment in combination with other treatments in colon and breast cancers, particularly breast cancers that have metastasized.
Does it cause liver problems?
Yes, it does. It does. Capecitabine. Gemcitabine. This is a very heavily used drug. So this again is an arabinose RSA prodrug. So this, it's a prodrug of cytarabine. You activate it by phosphorylation. It inhibits the synthesis of both DNA and RNA. In its first line treatment, you'll be combining it with other drugs, but you'll almost always include gemcitabine for pancreatic, metastatic breast cancer, bladder, ovarian, non-small cell carcinomas of the lung.
It's so crazy how much harm that anti-cancer drugs do to the rest of the body.
Yep. Decitabine is phosphorylated and put straight into DNA. It prevents methylation. It inhibits DNA methyltransferases because you can't methylate your DNA. Eventually you get abnormalities of gene expression that causes cell death. It can restore normal gene function and control cell proliferation, so it can actually cause tumors to differentiate and stop growing. So it's used for myelodysplastic syndrome. This drug is used for the syndrome that actually killed my mother. That's the decitabine. Notice again, it is a arabinose. Fludarabine. Fludarabine. So here's your ribose, there's your fluorine, and that looks a bit like adenine, but you've got this fluorine here, fludarabine. That's an antimetabolite. It's a prodrug. It's dephosphorylated to F-ATP. It enters a cancer cell, and then it gets incorporated into DNA strand, halts all lengthening of DNA strands, used to treat refractory chronic lymphocytic and chronic B-cell leukemias, not Hodgkin's lymphomas and T-cell lymphomas. Fludarabine.
For the exam, would it be beneficial to know then which drugs treat which cancers?
No. Just so I want you to recognize these drugs. And if you're nursing students, then you'd have to know what things you have to watch for, what tests you have to run, how to read the labs, all that. You're not nursing students. I'm more interested in the biochemistry,
So
that
just-
you know it's DNA.
So
it
what
inhibits-
just what the drug does.
Yeah. Yeah. And you might be interested in this, that's why I included it. Dane wants to be a doctor, so he might want to do oncology. So I have no
just didn't
idea
know if
what,
that's something we should be learning.
yeah. Nalarabine, you can see, is a drug that's converted to RAG by adenosine deaminase. Nalarabine. And so this drug, as you can see, gets brought in and it will terminate DNA and RNA synthesis. And it is brought in by these nucleoside transporters, and it's very effective. 6-mercaptopurine. This is used as an immunosuppressive in people with Crohn's disease and ulcerative colitis. Also used against acute lymphocytic leukemias. When incorporated into DNA and RNA, those nucleic acids are useless. Useless. It inhibits the de novo synthesis of the purines. Mutations may lead to drug resistance. That's why you use it with some other drug as well. Combination treatments are the rule, usually. So the TIMP, as it's called, TIMP inhibits the conversion of inosine monophosphate to adenylosuccinate and then to xanthanylate. It inhibits these reactions. TIMP also causes feedback inhibition of the glutamine PRPP aminotransferase. So it inhibits de novo synthesis, inhibits the very first step of de novo synthesis too. So here's 6-mercaptopurine. You can see 6-mercaptopurine undergoes conversion by the hypoxanthine guanine phosphoribosyltransferase transferase. And then it gets converted to the diaguanine nucleotides. It'll cause cell death and what have you. And then TIMP, of course, will muck with these other things too and what have you. And yeah. Okay, we'll finish this up on Monday. Then we'll get into, we'll look at DNA structure. We'll get into synthesis of DNA. The enzymology of those polymerases is very interesting. And then we'll do a little bit on biochemistry of DNA and RNA and stuff like that. Then I want to get into a kind of special topics thing, looking at the control of appetite using hormones. Biochemically, it's quite interesting. The test you will take, the ACS test you take will.
Right, but I.
My final won't.
So it'll cover everything from the last exam.
Yes. Yes, exactly, exactly. So the exam day, and they asked me, my exam will not be cumulative. The ACX exam you take will be.
Okay.
The last day of lab. So you take the exam, then you go out and man your posters and yeah.
What?
Because it's the same day as the display, poster display.
Are we doing that?
Yeah,
For-
remember I told you, you were part of that.
I don't know what are we doing?
Your thesis, yeah,
So we're doing a poster presentation and a.
yeah, yeah. If you want to do your poster presentation on a computer and do a slideshow that,
So we could just shorten our.
Yeah.
presentation.
yeah,
Doing that one.
yeah.
That's a lot better.
I sent you the result of the lab, Dane. The Western lot worked. In fact, it worked quite well. If you look at the bottom of it, there's a nice band.
Mhm.
That's the ICS.
Okay.
Pulled down with the frataxin. So the co-precipitation worked and it worked quite well. I'll bet you if we'd looked at enterocalin, the enterocalase, it would have come down too. But we didn't look at that, so there we are.
Yeah.
Okay,
Okay. Well, I hopefully will see you on Monday, but if not,
okay.
I will keep you
Keep me
updated.
posted, brother.
Yep. All right. Sounds good.
Okay.
Yep. Bye.